Well, welcome everyone to this week's Safer World by Design webinar. I'm very happy to have a really nice panel with me today. And our theme is good and best practices. A lot of us, and I think yourselves, have been involved in research in biology and toxicology, developing new methods. When we come to transfer them to practice and really have impact, being used by industry and eventually, hopefully, regulatory acceptance, this aspect of good practices, best practices, to start them as early as possible, even during the research program, are a key factor for eventual success and impact. So that was our theme today. We're going to have a few short presentations on different aspects of that, starting with good cell culture practice, and then working through data, stem cell biology, toxicogenomics, and then finishing on one of the emerging methods with organ on a chip technologies. And then we'll have, you can be entering your questions in the question box or chat at any time. And we hope to have a really good question and answer session and panel discussion. We'll all come back on for a good bit of time at the end until we all exhaust ourselves. So I hope you find the session enlightening, interesting, and stimulating. And I'm very happy to start with Glenn Stacy. I was very happy to work with him on a couple of different research programs in the past, and he's going to start us off on the very important topic of good cell culture practice. So with that, I'm going to hand you over to Glenn. Take it away, Glenn.
Okay, thanks. I hopefully everybody can see the screen. So as Barry said, I'm going to talk about cell culture practice and the new version, which is about to be published this year. I'm a member of the GCCP2 steering committee, which has been run by the Centers for Alternatives to Animal Testing at Johns Hopkins and Constance Universities. I'm also the Chief Executive Officer for the International Stem Cell Banking Initiative, which brings pluripotent stem cell banks together to discuss best practice, and I work with the Chinese Academy of Sciences as well. So I'll get on with the presentation. Firstly, just to introduce the topic, good cell culture practice is made up of, in the form that's been published as presented here, as principles of best practice, not detailed protocols, but guidance for people coming into the area of cell culture work for the use of cells and tissues in culture. It was originally developed starting back in the late 1980s and early 1990s to address issues of irreproducibility in cell culture-based assays for safety testing, which were performed under quite stringent good laboratory practice GLP protocols, but still finding variation in the results due to cell culture-based variations. The first formal document that came out of this, the GCCP, was published by Sandra Kirker and colleagues in 2005 in LTEX. And following that, there was, a couple of years after that, there was a workshop at European Centre for Validation of Alternative Methods to establish what the principles of best practice would be for the culture of human embryonic stem cells. That's available on the ECVAM website. More recently, there's been encouragement from CAT and the OECD to develop task force workshops to develop a new version of good cell and tissue culture practice, which addresses the new developments in cell culture, which have taken this area by storm effectively and changed the way that we work quite significantly in many cases. It also deals with particular new topics such as establishing cell culture metrics, and this field has been traditionally has worked on non-quantifiable assessments typically. Also, it includes guidance on pluripotent stem cell culture, composite cultures with mixed cell types, 3D culture, microphysiological systems such as the organ on a chip systems, which are now representing more than 10 different organ types on a small chip. So I think before I go any further, it's probably worth just reflecting on the consequences of failing to adopt good practices like good cell culture practice. Probably one of the most common things is the unwitting use of the wrong cell line. This is a very old issue, and even back in only about 8 years after the publication of the very first human stem cell line HeLa, Steve O'Brien and colleagues were publishing on possible cross-contaminations which were occurring between cell labs, and this has continued as a discussion since then. And today, it threatens millions of dollars of research investment potentially. There's an assessment of that done by the ICLAC group by Friedman et al. In 2015, where they identified the very widespread use of cell lines which were known not to be from the original tissue. Also, there are problems that can occur with long-term passaging if the cells are not cryopreserved at some stage, genetic changes, loss of function, and accidental complete loss of cell lines. And another big issue is contamination, and particularly those silent or occult infections which are not immediately obvious when you're culturing a cell, such as mycoplasma contamination, which can rapidly spread through a laboratory, cause permanent genetic and physiological changes, and even corrupt human genome databases. And the publication by Langford et al. Showed that around 7% of human genome sequences on the systems had some form of mycoplasma genetic element to them due to contaminations. So these are big issues for the cost of research and for the accuracy of scientific data. So as consequences, there's a significant waste of time and money potentially, where experiments may need to be repeated once these issues have been discovered. There may be loss of key research materials due to contamination, and there may be lab shutdowns, and for some labs that can go on for months where unidentified sources of contamination have been found. Ultimately, of course, this also can affect scientific reputation through an impact on the quality and reproducibility of published data, and that can even lead to withdrawal of publications. And in the case of mycoplasma, that's occurred in hundreds of papers now where these have been withdrawn. So these principles, 6 principles of best practice in GCP are really quite important. Probably the most important of these is number 1, understanding the cell system that you're working with and what affects it. This is really fundamental to all of our work working with cell cultures. The second part is on quality assurance, firstly dealing with quality control measures needed for cell cultures, and secondly setting acceptance criteria for any cultures that are to be used in experimental work to promote their reproducibility. Thirdly, the document deals with documentation of cell culture work to enable replication of results by other groups, effective communication, etc. And there are templates for the kinds of data that, as a minimum, that should be recorded for cell and tissue cultures. And then in the latter part of the document, it deals with safe working practices and legal and ethical compliance norms, focusing mainly on the US and the European Union requirements. And then finally, something which is actually probably the most important thing in cell culture work, and that's training cell culture work, staff adequately in these principles and how to use cell cultures. So one of the things I mentioned was setting acceptance criteria, and I think this is a really important new element to the GCCP document that's now about to be published. There are key stages in developing these acceptance criteria, so identifying what the optimum state is that's desired for the cells that you're going to use in experimental work, using that knowledge to know how you can set the best parameters to measure, the best ways to measure the particular parameter quantitatively, ideally. And finally, what variation from the optimal state is acceptable. So what are your tolerances on those particular metrics? It's important to remember that whilst a good acceptance criteria are most significant for the final expanded cell culture to be used in an assay, they can also be extremely useful at different stages in the cell supply chain. So in cells that you're receiving from other labs, having acceptance criteria for those, having acceptance criteria for cells recovered from cryopreserved cell banks, etc. It's important to recognise the various content, which I'm afraid I don't have time to go into today, that's present in the new GCP document. And in the particular case of GCP acceptance criteria, it deals specifically with some of the specific issues relating to spatial and structural aspects of cell culture in 3D and other systems, the impact of the culture surface matrix, assessing the composite cultures and how we would describe those, the optimal conditions for those, and how we measure that. And then also cell engineering aspects and how we manage the production and quality control of cell engineered cells that have been engineered genetically. It's also important to remember that these acceptance criteria need to be periodically reviewed to keep them up to date with the developing scientific knowledge. But it's also important to recognise that we can't keep chopping and changing our test methods on too regular a basis without qualifying the methods we're moving into use. So careful validation of each new method is really important, particularly where diagnostic and regulatory purposes are in mind. I thought I'd also mention a few details that appear in the document relating to genetically modified cells. So these might be gene edited, they might be reporter cell lines, or they might be reprogrammed cells such as induced pluripotent stem cell lines. And there are a number of examples of considerations included in GCP for the inputs and objectives for the work in mind. Firstly, what are the critical molecular tools that are going to be used? What is the quality of the expression contracts, the constructs, the guide molecules, the enzymes that are used in making the cell system? Secondly, what cell substrates are suited to the proposed engineering technique? And this is now being experienced in the area of gene editing, where certain cell lines are much more robust or are much more affected by gene editing. And so selection of a candidate cell line for your gene editing work is quite important to know that it's going to work reliably. Thirdly, what criteria are required for modified cells? And that's really important to know what it is you're trying to make and what quality of material you need, and what are the qualities of the material you need. Secondly, GCP also includes examples of considerations for acceptance criteria for outputs, the cell systems. Where there are gene reporter systems in cell lines, do they correlate with endogenous expression? Is reporter expression affected adversely by in vitro change, such as cell differentiation and other changes in culture conditions? And finally, how effective are the on-off switches for inducible genes? Gene editing is also dealt with, and considerations would include: is the desired change completed as intended, and is it free of on-target mutations? Are there off-target effects at critical sites of the genome, which is more difficult to demonstrate if you're using a standard gene editing substrate? Then this experience can be gained with this over time. And thirdly, do unedited or abnormal cells persist in the culture? And genetically abnormal cell lines are quite often isolated in gene editing experiments. And finally, do the engineered cells that you've actually made retain their key features of the parental cells that you need in your new assay, or acquire the new features that you want in your new assay? So those are just some of the considerations that are included as part of GCP across the different sections. Finally, I just wanted to say a few words about assay development. So assay development is specifically excluded from GCP. GCP2 is only there to provide guidance on the establishment of the cell cultures that you'll put into an assay. That might include some differentiation in the case of stem cell based assays, where you have to differentiate the cells to make a more mature phenotype to use in the assay. However, there is a document that was produced by OECD in 2018 called Good In Vitro Method Practices. And this is a document which describes pretty much soup to nuts what you need to do to progress an assay towards validation. So this is quite a useful document if you're taking a research method and you want to apply it to a diagnostic or safety test using a cell culture. And GCCP2 will be an annex to give him once the GCCP2 final document is published. So in conclusion, I just wanted to reiterate that the key GCCP principle is understanding the cell culture systems you're working with and what affects them in terms of the environmental contamination and other aspects, passaging, etc. And that is quite a tall ask because we don't understand all of the functions of cell cultures and how they change. This is becoming increasingly important as cell culture systems become more complex, and there are new analytical methods emerging all the time, but we need to be careful how we apply them and make sure that we validated each new assay as we apply it. GCC2 provides a framework for assuring quality in current and developing cell technologies, and so is something that we can use moving into the future. Currently, a draft is available as supplementary data on the open access document by Pami's et al. It was published in Altex in 2020, and there's been a consultation period ongoing since then, and the final version should be made available once all of the comments have been incorporated. I think there were over 300 commenters providing input to this document since it was published. And then that will be produced as a final publication in 2021. So I'm happy to take any questions on this in the panel session, and thanks for your attention.
Well, thank you for that, Glenn. Just pointing out to people who have arrived that you can add your questions to this question box in the panel as we go, and we'll field those questions as best we can in the discussion part at the end. So I wanted to cover the topic of data as part of this best practices discussion, and with a focus on integrated testing and assessment based on combining different kinds of in silico and in vitro data, and doing that with good data management best practices that ensure data integrity. So as Anthony Fauci often commented on last year, sound decisions should be based on data. And my theme is it should be based on data with best practices in preparing it so as to ensure its integrity in its use as evidence supporting decisions. Now, if we go back about a year ago, many scientific groups were using their resources to rapidly disseminate information and findings, hopefully useful to COVID-19 research, and typically accommodated by many rapidly prepared preprints. I take here as an example the highly visible case of hydroxychloroquine and azithromycin that was published end of last March as an illustrative example. In March 2020, French researchers published a study on 80 patients claiming a repurposing therapy to be effective for treating COVID-19. Many aspects of the study had criticisms, including on data rigor and statistical analysis, but the results were disseminated prominently, including on Fox News and by the President of the United States at that time, and very quickly it moved from a preprint to a cure for COVID-19, but based on supporting scientific evidence and data that was at the very least controversial. Now, when you look at the preprints related to that story, there was data reported, but often, like I'm showing as an example here, as a table in a supplementary Word document. And many other preprints, if you look back at that time last spring, had these characteristics. often without supplementary information or data, or if information was provided, they were not according to what are called the FAIR principles, that data should be findable, accessible, interoperable, and reusable. So this has been a problem we've known about for a long time, but we have been relatively slow to address it. The problems, however, around that story were not just confined to preprints. As reported in June in Science, 2 elite medical journals, The Lancet and the New England Journal of Medicine, had retracted coronavirus papers over data integrity questions. On the other hand, perhaps you could say a positive, epidemiological data and models were shared with the public last year at an unprecedented level. I show here one example from our contribution using web-based data resources supporting the daily national reporting of COVID-19 data in Slovenia. And this sort of brings us to the core question. Can we ensure data integrity in scientific communications, and how? So although legal and confidentialities brings significant issues, there is much we can do to improve the quality and the integrity of data associated with our scientific communications, including, I would claim, even with rapidly published preprints. I'll just go back. Increasingly, we have been building consensus on approaches to data sharing within the community. As for example, shown here between European research projects in nanosafety and associated interactions with other international programs in the United States and Asia, currently progress within the Nanocommons Community Infrastructure Initiative. These efforts should help enable a sustainable and harmonized knowledge infrastructure supporting the risk assessment of nanotechnology. So we've developed and share here a tutorial, or a set of tutorials, I should say, where the user can work through the different steps in preparing and sharing data. And we use PCR as our example here as one of the most common biological experiments used in both research and testing. So to start with, we need to work through the preparation and processing of the raw data. The metadata needs to be prepared to describe the data set, and the data organized into a format carefully labeled by the metadata. The data can then be processed by software into a database. And this procedure can also be elaborated through the use of validation software to check the validity and completeness of future data sets, including updated versions of data sets. Once the data has been uploaded and validated, we can use the metadata and the data to provide access to it through well-defined instructions and information provided to application programming interfaces. And this can be used as a basis for many different kinds of services, tasks, and web applications. I show one example here on the screen using notebook technology linked to such database interfaces to describe the procedure, the data, and the results and their analysis in a web-based, cloud-based kind of manner. Now, because the metadata has been captured carefully in our preparation of it in the first step, when we go to report the experiment and the results, we can also give a much more comprehensive description of the experiment and the conditions associated with the data. We can also provide interactive data analysis to the data in a live form, which can be viewed on the web, and in which the user can interact and choose their way of looking at the data, which perhaps is different to what we might ourselves have thought of as interesting as the data generator. So the data is alive. Also, you can make changes to the code and the analysis can be updated interactively in real time. And most importantly, the code documents the procedure and the processing and analysis of the data, which is part of this integrity goal. Now, like many, we had a virtual conference on OpenTox last year, and with that conference, we initiated a collaborative internship program, and we've made this best practices a theme of the internship program. And as the interns are going to present their work on knowledge resources, they're going to use such practices and start presenting them in webinars on OpenTox starting this month. More recently, we had a second generation version of OpenTox we called OpenRiskNet, and there we had the use cases drive the development of the e-infrastructure. So the specifications, the development of the data modeling resources, how they talk to each other, how they exchange data with each other, and with integrity, with interoperability, with these FAIR principles, becomes a really important part of such an infrastructure working in a reliable manner. We initially proposed this approach over 10 years ago within OpenTox, and now it's developing, I would say, in a more mature way. And currently, we're planning to make such infrastructure, including data, coming from the European programs, available as part of the European Open Cloud initiative, which is a major initiative by the EU currently. A related initiative is our collaboration with regulators. So here we're working with the FDA and CTR group to carefully disseminate a curated data collection on androgen activity. It should support users in search of the data, but also users using interfaces to such a database to build predictive models, and also to make this data and models available on the web, as I showed in the tutorial example. And here's an example from a partner, PK Derm, where we would actually use the approach we showed in the tutorial to the presentation and sharing of new experimental data. Safer World by Design, we're developing a platform supporting integrated testing and assessment for the design of safer products. And for such a goal, the data management and analysis best practices I'm discussing here are a key aspect of our vision to provide a reliable platform supporting integration of evidence, whether it's decision-making in early product development or eventual use of such evidence in regulatory reporting. This aspect of data integrity, I think, is a really key part of being successful and having impact on these new approach methods, gaining wider adoption. So I provide an example from there on a relatively simple skin irritation assay. Again, because we've captured the metadata at the lab in the CRO, we're able to include that in our reporting. We're also able to provide details on the experiment, on how the experiment is organized on the plate, and the controls, the raw data, and all the way through then to processing of the data using then the web notebook technology, so the results can be returned to the user in such a way, a report, but with everything well documented and with the data. So on the EU ToxRisk program, we're currently working on such practices for supporting the development of new approach methods in safety assessment, to develop an approach to knowledge management on the program, but also to the longer term sustainability of the resources, that such data continues to be valuable for future consumption uses by the end users. And what maybe this slide brings out is you really need a well-organized set of processes on both raw data, process data, metadata, and protocols established on the program. And more, we're taking such approaches from program to program and into next projects. And I think it's a key aspect of ensuring the longer term value of the data generated on the program. Also, while the work is going on, the use of such methods supporting integrated analysis of data across different technology platforms, different kinds of assays and analysis approaches, it's a very valuable activity for supporting collaborative work, teamwork on different case studies, and also then to the eventual dissemination of the results to a broader audience outside the project. We can also use this kind of approach so long as enough attention has been paid to the preparation of the data to show integrated analysis, for example, on compounds or similar compounds across resources, so that the users can take a look at, as shown in this example, how does data for the compound I'm studying with regards to omics or assay data look like from existing data. And this is something they should be able to do quite quickly, and then combine it with similar tools on their project, and take a look at the combination of the data, the integrated analysis, again through a modern web-based way with the data being alive. So in summary, my theme was good data management practices, I think, are increasingly possible today. They are making progress. There's a lot of hard work still to do, but it's a worthwhile endeavor. And it's one which you can both work on, I think, productively, individually, and in collaboration. Thank you. So after that discussion on data, I'm going to now hand over to Andreas. Let me make you presenter, Andreas, who will cover stem cell biology. So Andreas, you should see a prompt on your screen to become presenter now.
Yes, I'm here.
Okay.
Can you see already?
Yes, very good.
Okay.
You can
Just scroll up.
You can go to full screen or whatever.
I think I go to full screen. Because I think I cannot see what's on the screen, but I hope you can see the presentation. So the topic I want to discuss is how stem cells are useful in biological systems, and the presentation will be more a general one. Not so
Andreas,
much.
sorry. So your slide is not showing here. It's the desktop which is shown now.
Okay, then I go back.
You just need to bring up your slides, Andreas, please.
So now?
Yeah, just make it large, yeah. Maybe slide presentation mode.
I can also open the slides. 1 second, this is a PDF. Takes the slide over to the other screen and okay. Is this better?
Now we're in a presentation mode. See it,
Yeah,
two screens. Maybe you can go up the middle here, right to it, next right to it. Yeah, open the options. No, in the middle option. Sorry.
Okay.
Now it's gone. Yeah, here again, Anzeige Einstellungen. Then you can choose the other option.
All right. So thank you very much.
Yeah, now it's okay. Now it's good.
Now, okay, right. Just try to hide this one here.
Okay, good. Now you can go.
I think I can go in. So sorry about this. Yeah, so as I said, I will discuss a little bit the use of stem cell biological systems as a topic, but more from a general point of view, how stem cells mediate between the donor of the cells and the user of the cells in terms of modeling basically the donor. And the next slide shows what I want to present is that biological systems, whatever these are challenged by personalization efforts. So we have about soon 10 billion people in the world. These are affected by more than 10 to the 5 diseases, which are mostly caused by environmental factors, but also by the about 10,000 known protein coding genes, and affect about 1,000 cell types in the human body and 100 organs. So if you want to model a population of this size, we need to, of course, select cohorts and see how we can do that in vitro. The next slide then goes into the induced pluripotent stem cells, which are basically copies on a cell level of persons. And most of you know, of course, how that works. A patient cell is reprogrammed into a pluripotent stem cell, which can then be developed into all organs of the human body. And these cells can then be used to model diseases, to cell replacement therapies, to drug testing, toxicity testing, and to biologization of engineered products, for example. So these are, in principle, twins of the donor. And this twinning on a biological level, but also on the data level, poses a number of challenges, which I will try to discuss. So we have about, in banks and pluripotent stem cell banks, about 10 to the 4, 10,000 iPS cell lines, which represent, of course, only a small fraction of the 10 billion population. But there are already a pretty good presentation of some of the important disease cohorts. And the number is steadily increasing, not necessarily in stem cell banks, but in many labs, because it's very easy to make these iPS cell lines. So what can be done with these cells is shown here. In principle, they can be gene edited to create more, for example, disease models, to investigate the effects of toxins, for example, to differentiate cells into all organs, and then combine all these data, as we just heard, by tools which have been developed by, for example, by Barry and others, to get a complete picture of what's happening to the cells. Of course, that all needs to be kind of framed into an ethics platform, which allows us to do all these experiments and to use all these data. And if we now go to International Stem Cell Bank, internationally acting stem cell banks, and an example is here only mentioned, it's the European Bank of IPS Alliance, which collects induced pluripotent stem cell from European countries and makes them available globally, and of course findable and visible. And these banks usually are connected with two, let's say, systems. One is the stem cell itself, which needs to be produced in a reliable QC environment. This has been discussed by Gul and Stacy before. The cells need to be having a unique identifier and need to be fair in a way, findable. And there's on the other side data attached to these cells, which are represented by donor data, so by the person, the data of the person who donated the material from which the cells been derived, and the data directly associated to the stem cell. And somehow in between is the application of the cells. So the applicant, the user, wants both. They want the cells and they want the data. And then they want to create biological twins in a petri dish to measure, for example, tox effects. And they want to maybe create digital twins of the person by using the data. And this puts some challenge to the bank itself, to the resource who distributes the bank, which is basically sitting in between the donor and the applicant. The donor wants to know increasingly what happens to my cells, and do I really want to have the cells used in applications which have not been even known when I donated my material. And the applicants, of course, want to make sure that the donor, on ethical grounds, agrees to the research being done. And some recent applications or use cases of stem cells appear to be quite critical for the donor. For example, what I just mentioned, the creation of biological and digital twins based on organs made from his or her cells, the generation of in vitro embryos, which are now quite advanced. These in vitro embryos, which are being created through several methods from induced pluripotent stem cells, are basically a reflection of the person donating the cells in an embryo stage. And there will be only a few years from now technologies available to implant these first, of course, in vitro, in an in vitro placenta environment, but you don't need to have much fantasy to think that further. And another example is the creation of cell bots, which are cell-based robots which can perform certain tasks, for example, in regenerative therapies within the body of other people. So these are just a few examples, and there are certainly more coming up. So it's a question for the banks faced on how can we mediate these future applications with the existing cells, with the existing donor consent, and still make it possible. And there are tools needed to basically go back to the donor and ask him, well, do you agree with this particular application? And the donor then says, yes, of course, I agree, or no, I don't want that. And also the donor wants to be empowered. They want to know more about what's happening to my cells, what's the information arriving from all these experiments done with it, and maybe I want to be informed about that. So this is another challenge which these banks are sitting in the middle of. And there are tools needed, and we are working on this kind of tools to create a framework for acting of stem cell banks in a global environment with different ethical frameworks, of course, how to mediate this application and let's say the will of the donor, of what happens to the cells. And one of these products is called ethical avatar, to predict what the donor would probably think about the application. And if this prediction is a categorical no, then the cells could probably not be used. But if it is a yes, then the donor can be approached and asked directly whether that's possible. The other side is to collect by the bank or by databases the biological or digital twin data themselves, to make them available for the donor of the particular cell. So this is just in a very broad way presentation of the challenges being posed to existing internationally acting banks of induced pluripotent stem cell lines. Thank you. I would like to finish with that, my presentation. And I think I need to get out of the system again.
Oh, okay, thank you, Andreas. I'll hand over to Thomas now. I make you presenter. And Thomas will cover the topic of toxicogenomics. And just go to your slideshow.
Okay.
No, there you go. Okay, off you go, Thomas.
Thank you. So we'll address some best practices in toxicogenomics. So first of all, the toxicogenomics, it's the detection of up and down-regulated genes after exposure to a chemical using Transgenic Technologies. This technique is widely applied in different application domains such as chemical screening, pharmaceutical, cosmetics, food, and so on, because they offer a lot of opportunity, such as mode of action analysis, generated data for data core chemicals, hazard identification, and of course, ultimately, serving as component of alternative drug evaluation aiming at reduce and refine animal testing. As for the toxicology, toxicogenomics relies with main issues, which is the reproducibility of the analysis and the reproducibility of the results. In 2014, Miller and colleagues published an article about how to improve reproducibility in toxicology. And according to authors, this to improve reproducibility, we need to adopt at several levels some best practices. The first level is the experimental one, where it's important to describe the experimentation to allow replication of studies. Next level, it's the think about the relevant technologies to answer our biological question. Then the statistical analysis, which need to be well described and also appropriate. And finally, data management question about how to keep our history of external data or databases used for the analysis. So in toxicogenomics, the first best practice to adopt is in the level of the model, or to define or to select the model for the analysis. Since several years now, we're moving from animal testing to alternative methodologies, but the in vitro approaches are challenged. We already know that it's due to uncertainty and limited transferability power to the in vivo model. So here we need to choose the relevant model that will allow us to answer the biological question. There is plenty of models such as tissue slices, primary hepatocytes, cell lines, organoids, and so on, and each of them came with some advantages and drawbacks. So we need to study which are the advantages and the drawbacks to define our model. Using this model, when we use this model, we need to keep in mind is to follow the existing guideline from the OCDE, for example, that provide us some best practices and allow us give to the analysis some reproducibility. Then when we define, when our model is defined, we need to select the relevant transcriptomic technologies. Among the widely used technology, we can use the microarray one, which is the cheapest technology, but restricted to the interrogation of a set of genes. New technologies which come from the single-cell RNA sequencing technology, the barcoding technology, which is called the free-prime RNA sequencing, which is one of the cheapest transcriptomic methods and allow to investigate the gene expression of all known genes in the cell genome. If we want to investigate for new genes detection or new transcript, that we could use the traditional bulk sequencing. And if our scientific question relies on the cell susceptibility to the exposure of a chemical, here the single-cell RNA sequencing, it's the relevant technology for our question. So, according Miller and colleagues, and we need to well describe each step, to well describe our biological question that help us to define the model and then to select the transcriptomic technologies. Then we will be able to prepare our samples, to expose the samples according guidelines, to prepare sequencing library, sequence our sample, and then we'll start the second critical part of the toxicogenomics analysis, which is the data analysis. Data analysis relies on analysis strategy, which is lab and team dependent, using several softwares, packages with different versions, work on specific environment, and need some external data such as genome annotation, ontologies, and all of them with their own version. So to make analysis strategy reproducible, we need to adopt some best practice that will allow us to share the workflow, to share the analysis strategy with our team, lab, or with our partners, but also to run again the analysis several months or years later. The first best practice to adopt in this point is to follow the microarray sequencing quality control, the MAC and SANE consortium advice, which currently driving the reproducibility of transcriptional measurements and give some advice about which fold change used or which p-value used, and more in general, which transcriptional analysis strategy to adopt. But there is a more technical point to make to improve reproducibility in toxicogenomics. It's to use some tools like workflow engines and control environments to improve the reproducibility. Briefly, workflow engines are some software that will link all the software used in your analysis strategy and allow you to analyze your data with only one command line. Then, combined with control environments such as Combat, Docker, or Singularity, you will able to improve reproducibility and easily share your workflow, your analysis strategy, and run again this analysis several months later. Another critical point in the analysis, toxicogenomics analysis, it's the data management. So as Barry told us, data management, it's a critical point, and we need to adopt some best practice and our folder structure or file naming to be able to understand and reuse the data several months or years later. And using this, we should be able to easily understand who has created the data, what is the content of the data, what methodology was used on the data, and this plus information from metadata should allow us to run again the analysis several years later. This was the main steps to the best practice in toxicogenomics, and I just want to talk about the bonus step, which is the fair data. Barry already told us also about fair data. Today it's important to make the data fair, which means findable, accessible, interpretable, and readable, to be able to include our datasets into a global dataset and be a part of big project using metadata to describe our data, but also make them accessible through dedicated repository and make them interoperable and reusable using the commonly used data format. So just to sum up, in toxicogenomics, as in toxicology, we need to adopt some best practices by while defining our scientific questions, the model, but also following the existing guidelines to apply on the chemical testing, choose the relevant technologies, follow the best practices in the transcriptomic field, work with the workflow engine and control environment to improve the reproducibility in this field, and of course, manage our fair data. Thank you.
Okay, Thomas, thank you very much for nice overview and important topic. And now we go on to our final topic. And I hand over to you, Wolfgang, on some of the emerging methods using organ on chip, which potentially have large impact on new approach methods in coming years, and your wisdom on that, where we stand. So over to you.
Great, thanks a lot, Barry. And hello, everyone. This is I'm Wolfgang Moritz, and I'm overseeing the early product development and external collaborations and IP at Vivo. And yeah, my talk is about a topic about the, let's say, most complex applications in vitro testing, meaning looking at organ-on-chip applications. And so on-chip applications really are very challenging in terms of quality assurance. And of course, this is given by the fact that mostly and most systems now available very advanced in vitro cultures are used. I mean, it doesn't make sense to, put maybe a simple cell line in 2D in such microchips. I mean, it's done, but usually there's much higher requirement in terms of biological relevance. So that's therefore while mostly scaffold or scaffold-free models are used, either spheroids or organoids or stem cell-derived organoids, or specific hydrogel cultures, or even transvers, which have mostly barrier systems covered. So this is on the biological side. So it's very complex because these are mostly multicellular systems, and in most cases either stem cell derived or based on primary cell cultures. On the other hand, we have quite some development in the last 20 years on microfabrication system or technologies, which have fostered actually the development of microphysiological systems, which allows to interconnect different organ systems under more physiological conditions, meaning there's a flow, there's actually also available, also the mechanical stresses like shear forces, also the connection or the possibility to connect these different organs for organ-organ interaction, organ-organ crosstalk studies. These systems are used either in safety assessment or also in efficacy testing, but meant also to be used in the different phases of the drug development pipeline, from early target discovery to validation to drug optimization or lead candidate optimization. So it's meant to cover all the different phases. But it's also possible due to the modularity of these systems. I mean, there are different test system and biological models, but then also a variety of technical solutions, as are pointed out here on the right side. So there are quite a few suppliers on the market, and of course these developments are ongoing, and of course they are also challenged basically with the quality to achieve high quality standards. I mean, the complexity, of course, combining of complex in vivo models and in vitro models and complex technical solutions, and is basically increasing the challenge in standardization exponentially, I guess. I mean, this is only my personal viewpoint, but I would say that only few of these systems have really gone through the let's say full development, meaning that they are fully tested, they have quite sound and robust applications, and even they are validated, externally validated. I mean, that's true for part of these systems, but not at all to all, and not to speak about let's say regulatory approval. I mean, some of these systems might be useful then also in the preclinical phase, but of course then it is still a long way for regulatory approval. I mean, part of the companies might strive for that, but other ones probably are focusing more on R&D in the non-regulated phase of drug development. So when we look at what's been I mean, at the moment, most of the activities focus really on standardization, and I guess this is still challenging when we had heard, when we models used actually based on stem cells. Stem cell alone is very difficult for standardization, and then these models to have them sort of, let's say, a reliable production and differentiation method, which then is reproducible and gives robust data at the end. So these are certainly challenges, but I guess this needs to be developed like any other previous in vitro system had to be developed and had to go through these phases. And it's clear that these guidance documents, which were nicely described previously from Glyn Stacy, I mean, the good cell culture practice or good laboratory practice, which are now annexed to the given document, are extremely helpful resources to tailor the even complex in vitro models to the high standards which are required in terms of data quality, but also in terms of scientific integrity. So these documents have all these aspects covered, which finally culminate in one of the most critical issues is the quality, basically, on the systems, aside from their field of use applicability. But I guess quality is really prime, especially for those complex systems. I want to just give you an example on what does that mean, best practice in organ-on-chip applications. And so I can only talk about our experience based on our microphysiological system, which is a very simple concept, basically. So we have, on the right side, you see this plate is basically a microchip but adheres to the SLS standard format. So we have basically 10 cavities which are interconnected by a channel, and on the right and on the left side we have a reservoir. So the flow is generated simply by a hydrostatic pressure, just by tilting up and down the plate. And this creates some flow and interconnection of the different organs. And the organs actually are mostly spheric-based organs based on primary cells. And we focus, as Insphero, as a company, focus pretty much on metabolic relevant organs. So it's delivered pancreatic islets in the area of disease modeling, but also tumor and cancer in the cancer research area. And these models actually are used for disease modeling on one side, but also for safety assessment. Now, since we use primary cells, so we have to go through a lengthy evaluation step of the different lots, donor lots, which are provided by the cell suppliers. But once they fulfill all these stringent criteria, we have quite large lots, quite a large number of vials available, so we can guarantee a certain consistency in quality and also performance over a few months or over a few years even. Okay, so the beauty of that the system is that it's actually the production of the microtissues is decoupled from the assembly of the chip. So basically, we can perform, before we transfer the tissues onto the chip, we can perform a QC. So we have our standard QC criteria, acceptance criteria. And once those are fulfilled, we perform the transfer onto the chip, which is already very extremely helpful, and it minimizes the risk actually for failure, because we already know what quality of tissues we implement onto the chip. So we can basically create in vivo-like functions, which is defined first of all, on one side by the microtissues, organotypic microtissues, with in vivo-like conditions, which is defined by the microfluidic system. Now, you see, this is an example of how we assess the quality of the tissue. So that's our production platform, either 96 well plate or 384 well plate. And we see nicely on these scans the homogeneity of the tissues. They all should have the same size. And also there shouldn't be any particles in there which could interfere with the microfluidic applications. So we see those on the right side, you see a basic diameter of these spheroids over 10 consecutive production runs, and this clearly demonstrates how reliable and reproducible the quality of these tissues are. And this is also signified by the ATP content. So we have roughly a 10% variation in the ATP content, which is a pretty good number, I guess. So that's the quality control which we can apply, and these acceptance criteria are also monitored over time. So that's part of the quality management process, as we are also ISO certified. And this foresees actually the monitoring of the production quality by KPIs, or key performance indicators. And we define the key performance indicators basically on few features basically is one is what I already described, is the average diameter or volume of the tissues. Then we have ATP content, and we also can apply functional readouts as a quality measure, which is for the example of pancreatic islets. We also look at the glucose stimulated insulin secretion. And so we have certain range of acceptance. The certain values need to be in that range. If we have values which are out of the range, then it's a basically out of specification case. And if that's the frequency is increased, I mean, we can accept one of these different parameters be out of specification range. But if that accumulates, then we have different levels of alerts. Well, first is the warning limit, which will eventually trigger an internal investigation. But if we have more of those warnings, then it will trigger even a CAPA investigation. So that's clearly defined, and so we can also guarantee and that we will identify trends to worsening of the quality, and yeah, take over immediate measures here. So that's part of the quality management system. And so we're also transparent. So all these data are going to be provided to the customer. If they order tissues from our side, or if we implement those on the Onchip platform, we use these quality data in order to decide whether they qualify for Onchip applications or not. So we first provide these scans, which you already have seen in a previous slide. But we also offer these quantitative quality data, as already mentioned. So it's clearly identifiable also for the customers in what range they are, whether they're out of specifications. If they're out of specification, then we accept that once. We also make a specific note about that. And of course, donor information is very important. As I said, I mean, most of our models are based on primary cells. So therefore, we need also to provide the donor information. Then the customers needs to be aware that whether it's still the same donor or whether the donor changed or not. And they can relate to the donor information and their previous data they obtained from previous production lots. So there's full trackability of the data, also of the production process, and we also have retained samples in order to monitor potential latent effects. Okay, that's basically already what I wanted to highlight and also point out. I mean, we've chosen definitely two totally different concept of microfluidic systems or Onchip systems. This one is really demonstrating that we have through the decoupling of the production process of the microtissues and the setup of the microfluidic system, we have an advantage in terms of quality assurance. The tissues can be QC tested and before the assembly, and therefore it reduces the risk of failure. So in general, the simplicity of the system warrants a certain stability and robustness, which is a key actually for Onchip applications. Well, that was it from my part, and I would like now to hand over to Baron Natak, who is actually senior scientist at Tissues, and she's going to present you a completely different Onchip model, which is much more complex, and also the related quality assurance measures they apply. So thanks a lot, and yeah, Baron, the stage is yours then. Huh?
Thank you, Wolfgang, and I'm handing over to you, Baron, now. Baron, I don't hear you right now for some reason.
Okay, how is it now?
Now we can hear you. So
Sorry.
start again.
Okay,
Okay.
from the beginning.
Go on. Yeah, it's all right. Go on.
Okay, thank you, Wolfgang, for the introduction. And he actually already mentioned quite a bit from what I would like to, as the basis of the tissue engineering, 3D tissue engineering, and switching to the direction of microphysiological systems, and gave a nice example of a plate-based system that they are using. And I will give you a different perspective in terms of multi-organ approach that we have. So here, in a nutshell, basically for an assay development, we put required organs together. That is interest. That would be the aim of the assay. Depending on how many organs need to interact, and it would be 2, 3, or 4 organ chip that contains the relevant amount of organs together. This could be tissue engineered organs, or this could be biopsies, so ex vivo tissue. And that allows us also to be flexible. So we also wanted to aim for a stable, to provide a stable source and be flexible. So maybe I will also just now give you a bit of our point of view to explain what we do, and then how we come to the point of quality points, quality control, and what we think is important in that sense. So when we established the multi-organ chip, we aimed for the organs to interact with each other in terms of showing the metabolism, and then seeing a metabolite effect on another organ. And for that, we actually provided a closed circulation, so that we established closed circulation in the chips. And also we observed the importance of having pulsatile flow that we would have as in the heart. So these were important developmental points for us when we were establishing the multi-organ chip, and to be obviously to be able to image the chips. So this is basically our main hypothesis based on for the organ interaction. So to have it flexible, if you need a standard size, if you need insert format, if you have spheroids. So this is also something we work on, we provide. But imagining that this is a very new field, that we also need to be very flexible to develop any new approach for the for a required assay. So what we do is rapid in-house prototyping. So we can do it. We can do a new type of chip when it is needed in-house, and then it is important then also to produce the chips in-house to have the quality checks. We have a dedicated manufacturing environment, and then we have many quality assurance steps to provide a functional system. Whenever we have a new microfluidic setting, we always, just an example, we always characterize it particle image velocimetry in this case. But so imagine that having we have a control unit that actually provides the pulsatile flow to the chips, and then we have the chips that we also need to quality control. It is important to provide the functionality of the equipment and their safety. So basically, just an example of a document. I just took a snapshot that a control unit release protocol is like 35 pages long of checklist. And so for the chips, they have to really provide some system assurance and functionality to be able to show a reliable results. So with that in mind, so to put these together, basically, how we see as a reliable approach would start with qualified equipment. For the devices, all the disposables, accessories. So once we have the qualified equipment, we basically, the next level is the biological model development. This is this is usually in the academic level, that when the organs are, or the engineered tissue or whatever approaches, has to be tested first. This is usually the proof of concept level. And then once we have the proof of concept with the equipment, the first results, we can basically, we move on to produce more data for a qualified biological model. And this is established. So the established model becomes an assay, and this is the part where we start to use it in a context. And the next level would be to have a qualified assay. So with the data we collect, we reach the point of qualified assay, and this is the moment where most of the pharma partners that are starting to use these assays for decision making. So so this is what we believe in our point of view. These are the minimal requirements in a GCCP standard to have a qualified assay to be able to make a decision making point or decision making assay. And this brings us closer to a validated, a regulatory approved assay. Obviously, we are just, coming closer. So where we are with these is that we have assays that already reached level 3, so like qualified assays that are already in use, and more are on the way in the pipeline, that in the level of academic or qualified models. So that basically is, from our point of view, what are the minimal requirements, and I would be happy to answer your questions.